Welcome to the TWIML AI podcast. All right, everyone. I am on the line with Jianxu Chen. Jianxu is a scientist in the Assay Development group at the Allen Institute for Cell Science. Jianxu, welcome to the TWIML AI podcast.
Hi, Sam.
Hey, it's great to speak to you and meet you, and I'm looking forward to our conversation. To get started, why don't you tell us a little bit about your background and how you came to work at the intersection of machine learning and biology?
Okay, I got my PhD in computer science in 2017 from University of Notre Dame. And during my PhD, I mainly conducted research in machine learning and image analysis, and and more importantly, and their applications in biology. So before I came to Notre Dame, I have no clue what I'm going to do in the future. And later, I just all of a sudden, like we're randomly came into a very fascinating problem in biology and trying to use computer science algorithm to solve it. And that is the moment I say, okay, oh my God, this is so fun and very impactful. So that's the place that, or that's the moment where I decide, okay, this is my career. And then after I graduated in 2017, I came to Allen Institute for Cell Science, where we have very heavy, like multi-team doing team science, where we have lots of biologists, computer scientists, computational biologists, and software engineer, and everyone work together to solve very big fundamental cell biology problems. And machine learning is playing a critical role that attracts me here.
When you first got started, were you more coming from a biology perspective or more from a computer science perspective?
I would say I came from more computer science perspective, and I always use a very interesting example. I would say, like before 2017, I know nothing about mitochondria. I don't even know that particular English word, and I don't even know what does it mean. So that's really funny. But right now, I'm doing lots of segmentation analysis, interpretation on mitochondria. So that's really something really funny to me, actually.
How did you approach that? A lot of folks that are doing machine learning, don't have deep experience in a domain and have to get up to speed in the domain. And the domain that you are getting up to speed in is one that is particularly complex, which has its own many open questions around the biology. How did you approach coming up to speed in it?
Yes, that is fantastic question. So actually, I will probably cover more. I cover a lot in that particular question in my GTC talk. So the basic idea is that in computer science, we try to design an abstract problem. Say we have a very practical problem, like in a biology, in medical domain, or in whatever. The problem in our real life is more complicated. But computer scientists sometimes may want to start with a simplified version or abstract version.
Mhm.
And starting from there, we develop method, we develop all the kind of deep learning models and artificial intelligence techniques put in there. But later, I realized that how much we want to do this simplification or abstract is actually a key when we want to put artificial intelligence into biology. So sometimes the problem is oversimplified. That's the problem. That is the place I find myself really struggling with, and I was struggling a lot when I do the transition from a computer scientist to like half and half. So the strategy I took to do this transition is going back and forth between the knowledge, the biologist, and come back to my problem, and come back forth many times. When I show my result to biologists, I try to understand how they see my result. And
Mm.
sometimes, or most of the time, they see the result, or they see the image, or they see the figures, the numbers completely different from what I saw it. So they are
Mhm.
viewing it from a very different aspect. And by analyzing or by learning all such discrepancy between how I view the problem and how they view the problem, that's the strategy I took to transition myself from computer scientist to a like half computer scientist, half biologist.
That's awesome. Does any particular example of that kind of interaction come to mind where, you presented some results and they really saw them very differently from how you did and you had to kind of close that gap?
Yes, absolutely. There are tons of cool examples there. A very simple example is when we know lots of segmentation. Say the basic idea of segmentation is the computer reads in an image. We want to find or we want to outline the structure in this image. And then we can quantify the size of it or how long it is, something like that, right? So
Mhm.
the problem is pretty straightforward. So for computer scientists, I will just say, I will do like this and that method and extract object. I will how good is my output or how good is my algorithm? I will do that visually
Mhm.
by a visual assessment or some kind of comparison between my result and what I see in the image. So this is my understanding as a computer scientist. But when I go to the biologist, they are seeing that in a different way. So a key message is what you see in the image may not be the truth. You have
Mm.
to take the microscope effect into account. When you shoot a light to light up a small ball under the microscope, we are talking about spinning disk confocal microscope, where some particular cellular structure is labeled by some kind of fluorescent protein, and then it will be light up under the microscope and by the laser power. But what you see in the image is actually you have to take into account the blurry effect of the microscope because of all those optical details that I have no idea to understand at the beginning. So you see a ball with, for example, like 10 pixel wide. The actual object may be only 5 pixel wide, something like that. What you see is not the truth. That's what drives me crazy, right?
Mm.
So I have to understand what the optical details of that particular microscope and all those fundamental biology. Say, for example, the things cannot be like having two branches. It has to be a single filament, things like that. I have to combine my knowledge in computer science, also my the new things I'm learning about optics and also the new things I'm learning about the biology, I glue these three pieces together and reach a new, more accurate solution.
That's great. And I think that example is one that you're going to come back to when I look through your slides from GTC. A lot of the work that you're doing in the Cell Explorer Toolkit, which we'll be talking about, look like trying to kind of close this gap computationally between what you might see in one image and what's really there, but you're not seeing it due to some of these artifacts and effects that you're describing.
Exactly.
Is
Yes.
that right?
Yes.
Great. So maybe let's then use that as an opportunity to transition and talk a little bit about the Cell Explorer Toolkit and what is it really trying to do?
Yeah, sure. So at Allen Institute of Cell Science, we are trying to build this concept called Allen Cell Explorer Toolkit, which is a combination of cell creator, cell image generator, cell image analyzer, cell image visualizer, and cell image simulator. And in my GTC talk, I focus mostly on three parts: the generator, analyzer, and the visualizer, where the computation or the GPU computing is heavily used there. So the generator, if I want to describe it with one sentence, is like how we get a image to be analyzed. So apparently we need to get image from microscope, but with the help of GPU computing, we can generate more image from one single experiment. So that's the key idea of generator. The analyzer is, as the name indicates, is just given this image, how we analyze them. And finally, visualization is a big part of cell biology. So and also for GPU computing is certainly a big playing a critical role in the like modern visualization tools.
Mm-hmm. And taking a step back, what's the goal for the Cell Explorer Toolkit? Is this an internal tool that's used at the Allen Center, or is it designed to be used by external folks? And who are the main users? Presumably biologists, or is it something different?
Yeah, great. That's a perfect question. So actually, at Allen Institute for Cell Science, we are doing open science, and we want to make the tool that we develop here to be usable for everyone outside this institute. And the reason we believe our tool will be useful is that we are doing academic research at the industrial scale. By doing research at this level or at this scale, we may realize something. We hope to build some tools that will be more general or more stable or more user friendly for a broader audience so that everyone can use it. So that's the basic idea of why we want to build this toolkit.
And so you mentioned that you focused on these three areas, starting with the cell image generator. Tell us a little bit more about that and where machine learning comes into play.
Okay, cell image generator. Of course, again, as I just mentioned, we need the microscope to get some image. However, with the help of GPU computing, we can break some limit where the traditional microscope has. For example, in my talk, I showed an example where I took the image as a very low resolution, which can help me get overview of the whole colony, which is like maybe 500 of cells in the same image. But each of them has like relatively like not that detailed because of the resolution.
Hmm.
However, if we change some settings in the microscope, we switch to a different objective or using a different modality, the same colony, we can get it in a very, very high resolution. Say, for example, we may sacrifice something because, for example, we can only see maybe 10 cells in that particular image, but each cell has lots of details, which contains lots of meaningful biological information in there.
Hmm.
But there's a trade-off. So you cannot get both things at the same time. So you want either get more cells but less details, or get less cells but more details, right? There's always a trade-off. So
Hmm.
we are thinking that whether a computational model can help. So that's what motivates us to design this, what we call the transfer function. The basic idea is we build a deep learning model trained on some low-res image and high-res image pair. So whenever in the real image acquisition, we got like a sequence of low quality or low-res image, and which give us like 500 cells in each time step.
Hmm.
And even though there's not that much detail, but after applying this fully trained transfer function model on these low resolution images, we can like make it up and we can improve the quality or improve the resolution of each image so that every single cell we can put back all the details into the of each cell into that low res image. In other words, we are seeing high resolution of each cell at a very large colony, or it's a combination of larger field of view, or maybe seeing more cells in the image and
Right.
also seeing more details in the image. So
I'm envi-
traditionally, yeah, in the microscope, we cannot do that. But combining that with a computational model, we can achieve that to a very, very high accuracy.
I'm envisioning something along the lines of applying style transfer or deoldify, kind of one of these image coloring techniques, to the low resolution images based on the data from the high resolution portion of those images.
Yes. So the underlying techniques is pretty much the same as style transfer was the like common models people use in deep learning and computer science for style transfer, for super resolution, things like that. But
Hmm.
later, at the beginning, I thought, yeah, I have the exact same feeling. I think if people can do that on natural scene image, I think it's just a piece of cake and just put it on the microscope image. There's not a big deal. But later, after doing more and more validation, I realized one important piece. In the natural scene image, say you are trying to superize your image of a cat, right? So in your output, the supervised image, if the cat, the eyes of the cat looks a little bit larger or smaller, it doesn't really matter. Maybe 1 pixel larger or 2 pixels smaller, you may not even notice that, right? That they're still a cat. But for cell, that's different. That will be a very totally different story.
Yeah, yeah, I was going to ask that. I mean, a lot of, when I think about those kind of techniques, I think of generative models that,
Mhm.
in a lot of ways are just making stuff up. But if you're trying to use these images for scientific purposes, you actually want them to reflect the reality of what's happening in that cell population, not just, a visual high resolution thing that looks nice.
Yes, so that give us two things. First of all, it give us some direction about how we can improve our model on top of existing style transfer models. On the other
Mhm.
hand, it give us some hint on how we should validate our result and how much we should trust our generated image.
So why don't you go into those two in more detail?
Yes, for sure. So for the first part, so after seeing this particular thing about whether the prediction is to be larger or smaller and whether how much it affects, it drives us going back to the model, see what can we do to on the model to make this prediction more accurate, or what's wrong with the model. So then we realize that in our training data, or the way we collect the pair of low-res image and high-res image, it's very, very hard to get a fully aligned low-res and high-res pair. Think about how we capture the image in the real life. Say in microscope, when we capture a 3D image, you can think of like if you have a piece of meat, you want to slice it, right? So in the low-res image, the same piece of meat, you may slice it like at 3 different positions. But in a high-res image, you may slice that piece of meat like at 10 different positions, much denser, much smaller gap between each step.
Mhm.
So in that case, and also the position you do the slicing or you take the slice uh, may not be they may not at the same place. So anyhow, the low res image and high res image, they may misalign in Z. That is a fundamental issue.
Right, so you can't necessarily, directly match your target image or label image in the high res space to the low res space because they're misaligned in
Yeah,
terms of these slices.
correct. So we are trying two different ways. First of all, well, we modify the model, which we embedded a spatial transformer network into the model to learn such misalignment and try to correct it. That's one thing we tried. The other thing we tried is we try to develop a registration algorithm which can make this alignment to its best. Maybe they are not perfect, but as much as we can using a traditional image registration algorithm to the line image. And we tried both, and each has pros and cons. But the good news is both methods are improving the quality of the prediction by a lot.
I'm not sure if the image that I'm thinking of is one that corresponds to this point you're making, but there's an initial reaction I had to that you've got these additional slices and you're not able to align your slices, and so the approach is maybe one of interpolation. But there's this one image in there where that's showing that actually these different slices have totally different things in them, and so you can't
Mhm.
necessarily statistical interpolation of some sort probably isn't going to work very well.
So in terms of the slice, so I'm not sure whether I explained that clearly, but if I will try to put it in a different way. So if you think about the low-res image have 3 different slices on the same
Hmm.
piece of meat. On the high-res image, you have 10 different slices on the same piece of meat. So the first step, we need to upsample or do some interpolation on low-res image so that it will have 10 slices. So now on both images, we have 10 slices, right? So
Hmm.
now the number of slices match, but
Okay.
the first slice in the upsampled or the interpolated version, the first slice of that may correspond to the second slice of the true like high-res slice,
And that's
right?
where you're doing your registration.
Yes, that's what we are doing.
Got it.
Either we want to shift them a little bit and at the beginning, or we also develop a model that can handle that inside the model.
Okay. I think the image that I was thinking of is referring to something else. It was talking about predicted channels, and it has like each of the channels has very different things on it. And I was
Mhm.
interpreting that as slices, but that's probably something different.
Oh, by slice, I mean the slice along z direction.
Yeah. So you're doing the registration, which allows you to then use your upsampled image as a label. Is that essentially right?
So use my upsampled image as the input.
As the input.
Use the
Right,
true
right, right.
high-res image as my target.
Cool. So that was the first part, correct? And then the second piece is what?
The second part of actually, there's a second part of the image generator, which we call the label-free method. So what we have been talking about is from low res to high res, but there's another type of generation. Uh, so think about now we come to multi-channel is what you are picturing a moment ago. So
Okay.
yeah, so usually when we collect cell images, we will have multiple channels. Say we should light at the light spectrum. We have different type of light, and then we can have different channels as they have different dye in there. So
Mhm.
usually we will have a image with some dye to light up the nucleus, and some kind of protein to light up the one of the intercellular structure, and another type of dye to light up the cell boundary. And there's another special channel which is from the transmitted light, which give us what we call the bright field image. The bright field image is different from what we talk about in the other three, which require some fluorescent tagging, some fluorescent imaging. So the bright field image is just transmitted light. You just shoot the light there, you see the image without any harmful or photo any like dye that may damage your cell. So the light will not damage your cell. So in that sense, um, this is a general description of what we have in general. So I mentioned that there's a specific channel tagging only one intercellular structures because
Mhm.
that is a special cell line which we did lots of whole bunch of gene editing so that that particular cell can have that property where that particular structure can be light up with like the type of experiment. Each experiment can only light up one intercellular structures. And sometimes with very, very special gene editing techniques, we may light up two or at most three. Going beyond three will be super, super hard. So basically, we can see three structures at a time, at most, in the real experiment. However, we always have the bright field as a reference. The bright field image, I mean, the image we collect by shooting the transmitted light. So that is something harmless and can be cheaply acquired. Uh, so what we are thinking is whether we can predict the special intercellular structure from the bright field image, right? So if we can do that by given any single bright field image, we can predict 20 different structures at the same time for the same cell.
And in a non-destructive way.
Yes, that is the most important part, or it's a brand new way of designing your assay, your experimental assay. So before, we can only tag one structure at a time. And you can now see how these 20 structures live together in the same cell. You will never see that because every time you can only see one structure or two, right? So now with this particular technique, with any single bright field image, we can predict a lot of different structures for the same cell. That is a fundamental improvement in imaging or how we generate images. So that will allow us to study the cell as a whole, as like with all the different parts playing together, not individually.
And so in both of these cases, kind of going back to the earlier comments around the kind of the generative nature of some of these techniques, how do you measure performance and compare these cells that you're generating to actual cells in a way that ensures that they are high fidelity to what's actually there?
Yeah, that's a good question. So um, the strategy we are taking is application specific validation. Say you are carrying a, if you are, for example, when we talk about the label free prediction, say we are predicting 10 different structures from the same bright field image and how good they are, how much can we trust them? And and if we are talking about some application where we require the absolute accuracy, say whether the pixel-wise accuracy is exactly the same, matching the truth, then probably in the 10 structure, 9 of them is not that trustable in that sense. Because the pixel-wise accuracy may not be that high for some structures. So maybe one structure will have very high pixel-wise accuracy. But in other applications where we care about how different parts correlated to each other, like in the space, we don't actually need that much pixel-wise accuracy. What we care more about is whether they are predicting the overall position of that particular structure in the correct place. So with that in place, we can study how different structures functioning together, or whether there's any correlation with the structures. So anyway, that is what the strategy we call, like, we care more is like whether this result is suitable for that applications. So this is our strategy.
So, kind of taking a application by application approach to evaluating performance, some of which are concerned about absolute characteristics like size, and others are more concerned with relative characteristics like position.
Exactly.
That is the image generator
Mhm.
component. The next one you mentioned is the cell image analyzer. What is that piece trying to do, and where how have you used ML in that component?
Yeah, for image analyzer, in lots of cases when we've got image where we want to analyze it, the very first step is to do a segmentation. We just like binarize the image, generate a binary mask, indicate where are the structures, where are the intercellular structures. So that's the very
Mhm.
first step. We call it segmentation. So doing segmentation in microscopy image or microscopy image of cells is actually different from what we are doing, like the semantic segmentation or instant segmentation or all sorts of segmentation in computer vision, um, in natural scene image. That could be very different. So that difference comes from a couple of different sources. Let's start with a very simple example. Lots of people may know that for deep learning, people can just manually draw the boundary of that object. So no matter it's a people or cat or dog, we just manually draw the boundary of that. And we draw that boundary for, say, 500 images or 5,000 images.
Mhm.
And that may not be that hard. It maybe takes some time, but it's not that hard. That's possible, right?
Yeah.
After have this 5,000 image or even more, we just throw it into a UNet or some kind of ResNet or some deep neural network, and it will predict the mask for us.
Mm-hmm.
That's what like a lot of existing work are doing. But in microscopic images, especially for cellular structures, if you think about how complicated the topology or morphology of the structure could be, then you immediately appreciate that it is not possible to do this kind of manual annotation. So if you think about
I
some
mean,
structure,
I think when you think about, bounding boxes and how tightly packed these cells
Mhm.
are in some cases, it's clear that that won't work. But then you have pixel masks that are manually applied and other techniques that could conceivably be used.
for the pixel mask, even that could be hard if you think about in 3D. By the way, I
Mm.
forgot to mention that everything what we are doing is in 3D.
Okay.
So yeah, if you are thinking about a ball in 3D, and if you ask me to draw the mask pixel wise for that particular ball in 3D, I think I can do a pretty good job.
Mhm.
But if you give me a tree or a more complicated structure where different part interacting with different other parts, something like that, or a ball of yarn, you think about how they tangle up, right?
Mm,
And
mhm.
I want you to draw the outline of each single piece of yarn goes through the ball.
Yeah, it gets a lot harder in 3D
Yeah,
for
it's
sure.
not possible. Right? So the strategy we are approaching that is what we call which we implemented in what we call the segmenter, is we use some classic method. There are like more than 20 years of study of classic image processing techniques. They are still very usable, and they can give us a very, very good result to start. So we make a collection of a classic algorithm to give us some quick start. So based on that, for example, if you think about the ball of yarn, we design some classic algorithm can give us a rough segmentation of each piece of yarn. Mostly like not very accurate, but give us a lot of good segmentation here and there. So instead of manually draw where each piece of yarn is, we use a different way to do the annotation. We draw a bounding box, say here, this area, what the classic algorithm is doing is good. Let's confirm that. And then for that particular area, what the classic algorithm is doing is bad. So throw that away. So by doing this kind of curation, we say here are good, here are bad, here are good, and there are bad. When we may have a small amount of good segmentation and use that as our initial training data for our to train our deep learning model. And then you
Mhm.
have this model one. You apply it on your data, and then the result will be better. Now let's do again. Here is doing a good job. Here is doing a not that good. Throw it away. Here we are doing a good job, and there we are doing a good job. Okay, now we have our second round of iteration. We collect a larger set of good segmentation. Then we use that to train our second model. Now we keep this iteration going like iteration by iteration. So every iteration, your model is improving a little bit, more or less. So we hope to improve our model throughout this like different iterations, and at the end, finally, we will achieve a model that we can never achieve by collecting manual annotation.
What I'm hearing is that I know it's not quite the same, but it's making me think a little bit of like a active learning type of a scenario where you've got
Exactly. Yes.
you're trying to feed back to the system the training data that has struggled with the most, and in this case, you're using human annotators, but not to identify the actual features in your image, but rather to identify where the where your segmentation algorithm, didn't do a good job.
Correct. So that is a big part of the segmenter. There's another complementary part where you think about if you have two types of cells in your image, say mitotic cell and interphase cell. They are in different cell cycle and they may show up as different morphology. So one single classic method will not be able to do a good job there, right? So we can use two. We use two methods. Each will give us a different segmentation version. Now we have the same image. We have segmentation version 1. We have segmentation version 2. Each version has its own advantage and disadvantage. Okay, now we just use our human annotator as follows. Say we circle out in cell 1, in this cell, we make a circle. Okay, in this cell, we use segmentation version 1. In that cell, we circle out, we use segmentation version 2. In that cell, we use segmentation version 1. In that cell, we use segmentation version 2. So by doing this kind of circling, we are creating a merged ground truth that can help us train the model. At the end, the model is able to segment both type of cells correctly, very
Single model or multiple models?
single model to deal with two different type of cells.
And so this is again resting on the
Segmenter.
the
Part of
segmenter, exactly.
part of the image analyzer. So a little bit like 1 minute or 2 minutes down the segmenter direction is sometimes we may think of segmentation is most of the analysis is about segmentation. Sometimes it's not. Sometimes we can do something on the original image. So that's another part of the analyzer. This is what we call the integrated cell, where we build a autoencoder on the original image, where we try to learn the underlying correlation between each part of the cells and how they function as a whole. And that's another part of the analyzer.
You mentioned the autoencoder part.
Yes.
Elaborate on what exactly that's doing.
Okay, sure. So I showed an example in my slides where, say, I see cell boundaries like this. I see DNA patterns like this. And then can we use these two pieces of information, just purely taking an image, to predict where the mitochondria should live? So there must be there might be a correlation between the position of the mitochondria conditioned on the shape and shape of the cell and the DNA. So this is how the autoencoder is trying to learn. So basically, I take this, I'm trying to reconstruct it, and if I'm able to reconstruct it, and there I learn throughout this process, I learn some correlation um, between the mitochondria and the shape of the cell and the position of the DNA, things like that. So by doing this, we are trying to model the relationship between the different structures. I'm not sure if I make it clear, but
once you've done that and you've got this hidden representation in your autoencoder, how are you using that?
yeah, that is actually will give us lots of that's something we want to try to make some biological interpretation. Say, what are the correlation and how should we interpret from a biological perspective? So that's something we are doing right now to give it a biological interpretation.
Got it. Got it. So that part is ongoing.
Yes.
Okay, cool. And then just quickly, the third part of the toolkit that you mentioned is the visualizer. How do folks use the visualizer? Is that just kind of a GUI that sits on some data structures and allows you to look at these images that you've generated, or what all is going on in there?
Yes. So we build this. I mean, at our institute, we build this software called Agave, and that uses the GPU technology to do the photorealistic lightening and shading on our 3D microscopy data. So The reason we care about making the visualization more realistic, and also including something like depth of the image and things like that to make it looking amazing. It's not more than looking amazing. Actually, by looking this amazing image, it gives us some feedback from biological perspective. So when you look at a particular colony, the traditional rendering method will give you a kind of messy visualization in 3D, especially in 3D, right? So you don't have a good first impression about the biological property of the colony. But with proper shading and lightening and all this, the ray tracing techniques that we are using, we make the image looking very clean and very easy to digest, so that biologists can have a better understanding what's going on inside the cell and inside this whole colony.
And we will include a link to the your slides from GTC on the show notes page, and I encourage folks to check it out. For this part in particular, if only because the image of you've got one mode in Agave that's called cinematographic path tracing, and the images do look spectacular.
Yes, this is from our animated cell team, so I love their image. They're very beautiful.
Cool. And is there a ML component to this work as well, or is it just kind of raw number crunching using the GPU?
So far, there's no ML yet, but we are thinking about further integration.
Well, very interesting stuff. Jianxu, thanks so much for taking the time to chat with us about what you're working on. Very cool.
Of course.
Any parting thoughts or words for folks that might be interested in exploring this area further?
I would say we find that all this like machine learning stuff and the GPU computing solutions sounds like it's much more than faster computation. It's much more than better visualization. It's actually making us thinking about or doing cell biology research in a completely new way. And we are exploring different possibilities down this road. And that's something we are doing now and keep pushing forward into different directions.
Well, once again, thanks so much.
Thank you.
All right, everyone, that's our show for today. For more information on today's show, visit twimlai.com/shows. As always, thanks so much for listening, and catch you next time.